Okay, so today we will be discussing sample problems, questions that are related to the lectures that were covered in week 4 of Pharmacognosy and Metabolic Engineering course. The first question is BIA biosynthetic genes are expressed in companion cells of the phloem tissue in opium poppy. Identify the correct pair of cells where BIA are synthesized and localized in opium poppy respectively. So the first option is sieve tubes and lattice-fires. Option B is companion cells and sieve tubes. Option C is phloem, parenchyma and lattice-fires. And option D is sieve tubes and trichomes. You can take a couple of minutes and then think about the question and then unmute and explain what you think would be the right answer here. Okay, so Swati, we have started discussing. So the first question that we're discussing here is about the BIA synthetic genes. We know that these are expressed in the companion cells of the phloem tissue. So now we need to know where are the alkaloids synthesized and localized in opium poppy. So four different options are given here. Sieve tubes and lattice-fires, option A. Companion cells and sieve tubes, option B. Phloem parenchyma and lattice-fires, option C. Sieve tubes and trichomes, option D. You can take a couple of minutes and then uh, unmute and share with us what you know about this question. Yeah, you can unmute.
I think it is option A or option B.
Okay.
And participles or companion cells and sieve tubes.
Sorry?
I think it is option A
Okay.
or option B.
Okay.
Uh, sieve tubes and latticeous or companion cells and sieve tubes.
Hmm. So, do you know what these things are? What is sieve tube? What is lattice-fire? What are companion cells? Do you know about them? You can tell us what you know about that.
Read it. Now I'm not recollecting exactly how to explain the
Oh,
deep tube companion cell like differs.
okay. So, one thing that we can agree on is that these enzymes are produced in a particular tissue, they react in a particular tissue, and the compounds are secreted. So, do we agree on this part, both Anvay and Swati, that all of this is not happening in the cytoplasm of a single tissue? Rather, the gene expression, protein production, then the enzymatic reaction, and finally the translocation of these benzyl isoquinoline alkaloids, it happens in different tissues. Do we all agree on that?
Yes.
Okay. Huh. So, I've taken this information about sieve cells and sieve tubes from a paper. The link is provided here. So, sieve cells have the sieve areas on both end and the side walls, whereas the sieve tubes have both sieve area and the sieve plate. The figures are given there. So, what we need to know here is where exactly is the synthesis happening. So, the synthesis happens in companion cells. Although, okay, so the genes are expressed here, which means that the transcription and hence the translation is happening there. Then these seed tubes are involved in transportation and the storage. Then later on, whenever or wherever these alkaloids are required, they will be specifically redirected to those tissues. So based on that, we can say that the synthesis happens in the companion cells and sieve tubes is where it is stored or localized temporarily. Lactosifiers are majorly involved in storing latex or rubbers, rubber products as secondary metabolites, but not the kind of secondary metabolites, that is alkaloids, that we are dealing with. So lactosifiers are those tissues where these rubbers could be stored, but it is not the place where alkaloids are stored. Right? Was that clear?
Okay.
Right. The next question is select the correct set of enzymes involved in the production of morphine and alkaloids starting from thebaine. So we need to know what thebaine is and chemically what all transformations are happening. So if we know that, we will be able to tell what is the right answer or what is the right sequence of enzymes in this process. You can take a couple of minutes and then we look at how this pathway flows from one particular substrate towards a product and the entire pathway. Shobhana Priya, we've started this week's problem questions. We are in the second question. So, in this question, we are looking at the correct set of enzymes that are involved in the production of morphine and alkaloids from thebaine. And four different options are given here. You can think about the pathway, how thebaine is converted into morphine. You can think about it chemically. What are all the chemical changes that happens to thebaine to produce these morphine and alkaloids? And that will help you tell us what is the set of enzymes required. So you can take a couple of minutes and then unmute and tell us what you know about this pathway. Okay. Okay. Does anyone want to try answering this question? Anvay, Swati? Okay, let's look at the pathway that might help you. So, this is how thebaine looks like. It is produced from reticulum. Now, the difference that's between thebaine and the next compound is that you have a OCH3 group, which is oxymethyl group here. You have an unsaturated double bond here. Now, this is being replaced by a COO group. So, that is a O demethylase. Okay, so we now can go to option C or D. Then this here you see that the double bond has shifted from this to this. This is a tautomeric nature which can happen spontaneously. So that gives us from thebaine to neopenone to codenone. Okay. Then what we have is this double bond O getting reduced to OH. So reduction is the process where you have an addition of hydrogen. So this double bond O becomes OH. So this is a reductase. So you have a codenone reductase. And again you have another oxymethyl group here. And that methyl is getting replaced with an hydroxyl group. So again you have a demethylase. So in total you need to have two demethylases and one reductase. Among the options here, option C has two demethylases and one reductase, which is why option C is right here. Was that clear? Okay, we'll go to the next question. The next question is CYP80B3 is a hydroxylase catalyzing an upstream step of BIA biosynthetic pathway. It has been found that CYP80B3 catalyzes a regulatory step of the morphine and alkaloid biosynthetic pathway. Select the most suitable options among the following that you hypothesize to get upon antisense expression of CYP80B3. And four different options are given here. Reduction of total alkaloids in the latex, no change in the total alkaloid content of the latex, increase in total alkaloid content of latex, reduction in alkaloid content synthesized in the early steps of the pathway. So, to start with here, first think about this question. Do any of you know what CYP is? It's a common set of proteins, enzymes. Swati or Anvay, do you know what the full form of CYP is?
It's cytochrome 450 enzymes.
That is for, yeah, CYP 450 is cytochrome P450, right? Yeah, here it's cytochrome protein. Yeah,
Cytochrome.
right, right. And do you know what antisense expression is?
the basic form.
To be able to answer this question,
I
we need,
think
yeah, sorry, yeah, go ahead.
you have asked here to get upon the antisense expression of CYP3B3. I think
Yes.
it will be reduction of the total alkaloid in the packet.
Okay. Right. That is okay. We can reach the answer, but do you know what antisense expression is? Because you need to know what is actually happening.
Uh,
Huh. So do you know what antisense expression is? Is that a method where you introduce something to the cell, some gene regulation expression
yes, yes,
changes?
yes, yes.
Right. Do you want to share what you know about that?
Little time. I'll just think.
Okay, I have an image here to show what antisense oligonucleotides can do. So, there are two ways majorly in which antisense oligonucleotides can work. So, antisense means it is opposite to the sense strand. A sense strand is the one which acts as template for your transcription. Basically, that is the strands. I mean, sorry, that is the strand that is being read and that codes for the genetic information. When we have a complementary DNA that can bind to the strand that is being read, it can work in two ways. The first way is even before it is being transcribed, or sorry, even before the protein is getting produced. So, in eukaryotes, you have this process called as splicing. What happens in splicing is that, unlike in prokaryotes, where genes are well defined with a complete stretch of nucleotides, in eukaryotes, you have a coding region followed by a non coding region, followed by a coding region, and so on. So, the mRNA that is being produced will be chopped into different pieces such that you have all the exons together, which can later be stitched to give you different isoforms of the same gene based on the order in which they're getting stitched. Now, that process is called splicing. Now, this antisense oligonucleotides can either bind during splicing itself. So, if they're binding during splicing, you have changes in the gene that will be later translated at the splicing level itself. Or once it is being spliced and then stitched, it's called a mature mRNA, the one that comes outside the nucleus into the cytoplasm. Now, again, this mature mRNA can also be bound to the antisense nucleotide. Depending on the region where it binds, it can either block translation at the beginning itself, thereby leading to no protein at all, or it can activate the expression of RNase H. Now, RNase H can specifically recognize DNA RNA hybrids. Now, this is an RNA. If the oligonucleotide that we use is a single stranded DNA that's complementary to our mature mRNA, that can be recognized by RNase H. And if RNase H recognizes this DNA RNA hybrid, it will chop it down. Thereby, again, the RNA will not be functional. So, this is antisense technology. Now the question is, what will happen if we use antisense expression of this cytochrome P83 80B3 gene? So for this, we need to know what CYP80B3 gene does. So they say that it's a hydroxylase that catalyzes upstream step of BIA biosynthetic pathway. Now, if it's going to catalyze an upstream step of the BIA biosynthetic pathway, it would mean that it would be responsible for the production of a precursor for the BIA biosynthetic pathway. So, if we block this particular enzyme which produces your precursor, then that would reduce the further downstream produced alkaloids. So, in that sense, whatever Swati mentioned as reduction of total alkaloids would be a right answer. But again, here we have two different options: reduction of total alkaloids in the latex, reduction of alkaloid content synthesized in the early steps of the pathway. So now both could mean the same, but there are some small differences. What this steps option D says is that when there is reduction of alkaloid content synthesized in the early steps of the pathway, it assumes that downstream there is no issue. But that would depend on other factors, other cofactors, and especially if your precursor is not synthesized, all the steps in the pathway would get affected. So, although people would consider option D as right, I would suggest that you clearly mention that option A, where all the total alkaloids gets reduced. That would be the most appropriate answer for this question. Right. Although we have mentioned that both option A and D are suitable, I would preferably go with option D here. Sorry, option A here. Right. The next question is, identify the correct set of novel benzyl isoquinoline alkaloids that get accumulated in Eschscholzia californica upon overexpression of scolerin O-methyl transferase. You can take a couple of minutes and then maybe share us what you know about this organism. We'll start with this organism, Eschscholzia californica. Okay. Hi Deepali. So we started with the questions for week 4 of Pharmacognosy and Metabolic Engineering course. We are in question number 4. So in this question, we are looking at the correct set of alkaloids that get accumulated in Eschscholzia californica when this scolerin O-methyl transferase is being overexpressed. You can take a couple of minutes, and before you get to the right answer, you can maybe tell us what the common name of this organism is. Eschscholzia californica. We can start from there. Then we can look at what this gene, scolerin O-methyl transferase, does and how its overexpression would produce or shift the flux from one particular metabolite to another particular set of metabolites. Okay, you can take a couple of minutes and then unmute. Yeah. So does anyone want to try answering this question? We can start with what the common name of this organism, Escherichia californica, is. Does anyone want to try? Okay. So Escherichia californica is California poppy. So this is also a, belongs to the family of Papaver somniferum, right? That's where you have your poppy. Now they say they ask for overexpression of scolerin O methyl transferase, which means scolerin is involved in this process. So, does anyone know where exactly scolerin is being produced? Where, as in it? Which step of the pathway? Swati, Deepali, Anbe? Anyone? Oh, okay. So, this is the pathway. Now, we have scolerin here. Scolerin is being produced from reticulin. We earlier saw reticulin in the thebaine block. Now, once we get scolerin from reticulin, scolerin O-methyl transferase, it produces tetrahydrocolumbamin. Okay, then it gets acted upon by another cytochrome P enzyme and gives rise to allocryptopene. That is one of the compounds. Allocryptopene can give rise to L-erythrine. Now, among the options we have here, we have allocryptopene and L-erythrine here, which is why option C is correct for this particular question. The next question is identify the benzyl isoquinoline alkaloid that got significantly enhanced in the latex of transgenic poppy expressing COR HPRNA. Four different options are given here: thebaine, codamine, laudanine, and reticulin. This is an interesting question. This is taken directly from a paper based on a particular study. We have references to that paper. You can check that paper once we finish discussing this question. So before we look at this, look at the right answer. Does anyone know what HPRNA is? Swati, Deepali, Anbe, anyone? HPRNA? You can give it a try because the more you interact, the more you can learn from this. That's the uh, point of this class. Okay, I'll tell what HpRNA is. HpRNA is hairpin RNA, and COR is an enzyme. So, in this paper published in Nature in 2004, they report on a particular strategy where they try to silence this reductase, codenone reductase, in opium poppy. So, here what they use is a hairpin RNA construct. Again, this is also a method that is based on RNA interference, where they want to do gene silencing. In this paper, they have designed multiple different hairpin RNA targeting the multiple gene codenone reductase family. And after they did this gene silencing, they measured how the profile of these alkaloids varied. So, codenone reductase occurs at this step. Okay, so they created this chimeric hairpin RNA that would help in reducing or suppressing the gene expression. And what they observed is that compared to the control transgenic latex, where you had morphine, codamine, oripavin, reticulin, and thebaine, with reticulin being the least detected compound here, that could be because of reticulin being the topmost precursor from which other compounds such as morphine, oripavin, codamine. So all of these are derivatives of reticulin. But when they introduced this COR HpRNA, which is the reductase hairpin RNA, they observed that this reticulin level increased significantly higher in comparison to the control. And the production of other compounds such as codamine or laudanine and all of that reduced. So, what was interesting is that a HpRNA that was aimed at reducing the expression levels of codenine reductase, in fact, reduced or in fact, improved or accumulated the levels of a compound that is 7 steps produced before. You have 1, 2, 3, 4, 5, 6, 7. So 7 steps. This is what is reported in the paper. So coming back to the question, so here they ask the alkaloid that is getting significantly enhanced. So that is reticulin. I hope this was clear. This is an interesting Nature paper that was published in 2004, 2005. You can check this. The next question is, which of the following amino acids serves as the precursor for benzyl isoquinoline alkaloids? And four different options are given here: tyrosine, tryptophan, alanine, and valine. You can take a couple of minutes and then unmute and tell us how this benzyl isoquinoline alkaloid pathway originates, where it originates from, and then we'll discuss further. Okay? Yes, Swati.
Is it tyrosine?
Yeah, you can. Okay. So you can tell us how from tyrosine it gets converted to other compounds. Uh, do you remember that, or do you have an understanding of what is the biotransformation that's happening?
Tyrosine to the codeine, often then again.
Sorry. Tyrosine to?
Actually, I'm traveling. I
Oh,
think tyrosine. Yeah.
yeah. Tyrosine is the answer. So it starts here. You have tyrosine that produces tyramine and then dopamine and so on to give thebaine, morphine, all of it. So yeah, so the root uh, metabolite or the precursor is tyrosine here. Right. The next question is correctly arrange pathway intermediates of reticulin biosynthetic route. So again we have, like we saw in the previous question, we know that the precursor is tyrosine. Now we should be able to tell how reticulin is produced from tyrosine. So again for this question, we need to be able to think what happens to these metabolites chemically. You can take a couple of minutes and then try answering this question. Okay, we look at the pathway. So from tyrosine, we get tyramine and then dopamine. From this, it starts. We get norcorcollorin. That is, that leads us to two options, either A or C. Tyrosine to norcorcollorin. Once we get norcorcollorin, we have a methyltransferase, and that gives us cochlorin. And once we get cochlorin, again we have another methyltransferase, now at the N position. Earlier it was an O methyltransferase, which would add a methyl group at the oxygen, making this hydroxyl group and O methoxy group. Next, we have a methyltransferase that would add a methyl to this imine group, giving an N-methyl. From this, then we get a hydroxy derivative of this N-methyl cochlorin, and then again we have an O-methyl transferase here. So this is the structure of reticulin. So this would mean you have tyrosine, norcochlorin, cochlorin, then methyl cochlorin. So that's why it's option A here. So one easy way to understand or remember these pathways is thinking about what is the chemical transformation that's happening here. O-methyl transferase followed by N-methyl transferase followed by hydroxylation followed by methyl transferase again. Okay. So think about these pathways in chemical terms. It would be easier to remember. The next question is identify the non-morphine and alkaloids from the options given below. Option A, thebaine. Option B, salutaridinol. Option C, neopenone. Option D, sanguinarine. You can take a couple of minutes and then think about this question and then unmute. Yes, sir. You can take a couple of minutes and then uh, tell us the answer. Yes, sir. You can unmute and tell us.
Um, I think option A and C, thebaine is a morphine alkaloid,
and C, neopinon also. So isn't morphine an alkaloid? Now between B and D, I think is the correct answer. I think is the option D, sanguinarin.
Right, right. Yeah, like you mentioned, we have already learned that thebaine is a part of your morphine and alkaloid. Now, thebaine can get converted to produce neopinon. So, thebaine to oripavin, now oripavin to neomorphinone. And this neomorphinone can also be produced by neopinon, which is directly produced by thebaine. So, these three options belong to your morphine and alkaloids. Now, coming to option B, which is salutaridinol. This salutaridinol, it is the precursor for thebaine. So since this is the precursor for thebaine, we need to consider this also as a morphine and alkaloid. Then the only option that is left is sanguinarin. But does anyone know to which class of secondary metabolites does sanguinarin belong to? If not for morphine and alkaloid,
Is it by pyrimidine alkaloid
why would you say that?
or
Any
pyrrolizine alkaloid? I'm not sure.
why would you say that? Any reasoning behind that? So this sanguinarin is produced from scolerin. Earlier we saw about reticulin and how dopamine from tyrosine. So sanguinarin belongs to the other class of these alkaloids that are produced from uh, tyrosine. It belongs to the same class as reticulin and scolerin. Yeah, so now you can tell me to which class of alkaloids does reticulin or scolerin belong to?
Tropane alkaloid?
Right.
Uh, yeah.
Yeah. Hmm. So we'll go to the next question. Which one of the following statements is not correct regarding BIA synthesis, biosynthesis in opium poppy? Uh, these genes are expressed in endodermis and pericycle. These are expressed in companion cells. These are assembled in companion cells and translocated to sieve elements. These are produced in sieve elements and then transported to lattice for storage. So, which of these is incorrect? You can take a couple of minutes. We earlier had another question that is related to this question. We had earlier looked at that. Maybe you can think about that question and try to solve this question. And take a couple of minutes and then unmute. Yes, Swati.
As we have discussed before,
Hmm.
the biosynthesis in opium poppy, the BIA biosynthetic genes were expressed in companion cell we have discussed before, even
Yes.
latticipers for storage and translocation to the sieve elements. So I think these all are true except A. A is the correct answer.
Right. Yeah. So since these BIA biosynthetic genes are expressed in companion cells and not in endodermis or pericycle, option A is the one that is incorrect regarding BIA biosynthesis. Fine. The next question is, which alkaloids were most abundantly obtained in top 1 mutant of poppies? Codenone and morphinone, thebaine and oripavin, codeine and morphine and heroin. You can take a couple of minutes and then we can start by discussing what top 1 is. So whoever wants to, so this is based on another 2004 study, I think, where mutations were made in one of these uh, enzymes. Okay, so top 1 is an enzyme. You can think about what top 1 could be, and then uh, you can maybe tell us the full form of top 1. We'll take it from there. Okay. Yeah, Swati. You can start by telling us what top 1 is.
Little bit, I forgot actually. Top one mutant of the poppies. I think codeine and morphine is the correct answer.
What is top 1? Can
Yes,
you think about top 1 once?
to your top one mutant. I read it, but means heard that video. But
Right.
now I'm outside. No, little. That's why it's not in top one. It's inhibiting of.
Okay. We'll ask others if they know. Others, would you like to try top 1?
Top one, what I remember from the video is was used for commercial production,
Right.
but to increase the yield. But I just now don't remember uh, which for what it is used. And the T states for some uh, name of the place, I think, sir. But again, I'm not able to recall that. So
Okay. You can. He could be Thebain.
top one.
I'll open paper here which had done this work.
Hmm.
I hope you're able to see. Are you able to see what I'm opening here on the other slide?
Yes, sir.
Okay, so this is
But
the
it's
nature
just
state.
dragging.
This
We
is,
can see the arrow. Hmm.
you can see, yeah. So here the top one mutant is Thebain oripavin poppy 1. So what they're trying to do here is that they would cut off this part and this part. So now what is happening is that compared to your control, which is shown as a spectra here, where you have morphine, codeine, and thebaine. You get codeine here, morphine here, and thebaine here. Codeine and morphine are derivatives of thebaine. We need to remember that. Now, in this top one mutant, this particular step and this particular step is cut off, which means that you will not be able to produce neopenone or morphine or codeine or any of this, but rather only theobromine and oripamine, which is what they observe in the spectra with top 1 mutant. Only oripamine and theobromine, which is also shown as a TLC here with the standards shown on the right. So in the left TLC, you see a band for 3 to 4 compounds, at least 3 clearly. The smaller band is corresponding to morphine, the second band to codeine, the last band to theobromine. But in the top one mutant, you see only for theobromine and a smaller band for oripamine, but no codeine and morphine. So this mutant basically removes conversion of thebaine and oripamine into other secondary metabolites. That is what was achieved. So now coming back to the question, this is also an interesting paper. You can check this out. The link is provided. So now here the question is which alkaloids were abundantly obtained in this top one mutant? This is again theobromine and oripamine. So yeah, so with this we have covered the questions that were set for today's class. If there are any additional questions or doubts, we can discuss. I will be available here.
So wanted to ask you the structures which are given,
Huh.
so many structures, how to like, is it required to remember? Because without structures, at least the alkaloids and all
Huh.
is okay. Like, yeah.
I would say again, you can remember what is happening chemically. You can remember just basic structures. I think you would be familiar with the structures of amino acids. So if you need to look at resources that tells you why a particular chemistry is suitable. Like for example, here you have from L-tyrosine to tyramine. This is a decarboxylation reaction which removes a COOH group. So if you are able to understand why tyramine needs to be produced and where it is produced, you will not have difficulty remembering the structure. You can derive it from L-tyrosine structure. And this way, if you are able to remember it as a sequence, this particular metabolite enters a particular tissue, and in that tissue, it has to be decarboxylated because of the pH or the nature of the environment. That would be the easiest way to remember because otherwise you can't remember all of these structures. Instead, knowing the key precursor structure and deriving the structure of your compound of interest from the precursor is a easier method. That's how we would have learned for other biological processes like glycolysis and so on. So you can apply the same logic here as well. That would be my suggestion, Swati.
Okay.
Right.
So what are the non-benzoisoquinoline alkaloids?
The last question is it? Or this question?
This one we answered that sanguinol,
Huh.
that non-benzoisoquinoline alkaloids, not yeah.
Wait, this is the top one mutant question. This is the non-morphine and alkaloid, which is sanguinarine.
Other than that,
I didn't quite get your question. Uh, you're asking for non-benzyl isoquinoline alcohol. You have multiple
Huh.
categories, right?
Examples.
You have, okay, let me, okay, I just did a quick search and I could get laudanocene, which was, I think, shown in one of the pathways. Laudanodine, benzyl isoquinone. I remember seeing it in one of the pathways. Yeah, this laudanocene, they call this a non-benzyl isoquinone alkaloid. This is called as, yeah, so they call this as a non-benzyl isoquinone alkaloid. And this, uh, what else?
Okay, sir.
Yeah, this is one. Uh, hmm,
Thank you, sir.
right, right. Deepali and Anbe, I'll be here. If you have any questions, you can ask me.
No, sir, no questions.
Okay,
Thank you.
thank you.
you.